All right, welcome everyone to our Applied Bioinformatics webinar. First of all, let me thank you for joining us today. My name is Tamás Kis. I work at the Semmelweis University Pediatric Center, and I'm the host of this webinar series. The webinar series is sponsored by the Data Science and Me LLC. I'm so excited to be here today and have an opportunity to introduce our speaker, Szonja Kovács. Szonja is a research fellow at the Oncology Biomarker Research Group at the Eötvös Loránd Research Network. She is not just an experienced user of a wide range of bioinformatics tools, but including Galaxy, but also she teaches bioinformatics at the Semmelweis University. The presentation will take approximately 35 minutes, and at the end of the presentation, we will give time for a Q&A session. The presentation will also be recorded and to make it available later online. Szonja, thank you for joining us today, and the stage is yours.
Thank you very much, Tamás, for this invitation and introduction itself. And thank you for these participants also to join us today. And I would like to start, if it's okay. So as the title should suggest, I will talk about the Galaxy platform, what is it good for, why you should use it. So first of all, what is Galaxy? I'm sure that for those who have already heard about it, know that it's a good data analysis platform for omics data, but is it really so single focused? Of course, the answer is no, because I asked it. Galaxy is like the many-faced god in Game of Thrones. It can be used for many other options. You can use for data management. You can handle, convert, share, edit your data, whether it's biological or non-biological. You can share your data and, of course, analyze it. You can use many visualization tools in the Galaxy platform. It has a really good community, and there are a lot of experts from who you can learn or help you. And it's a great tool for publications. So I would rather say that it's not only for a platform for omics data, but it's a bioinformatics or scientific workflow management system. I know it sounds really fancy, but it just basically means that you can use it without a programming knowledge, with a graphical user interface to many goals or tools. So in the Galaxy, you can use it for omics data, for ecology, climate modeling, imaging, statistics, or machine learning. And there are tools which you can use for cheminformatics also. And why you should use it? Because of many reasons, but to tell you that it's free, it's open source, it's available from everywhere in the world, it's web-based. You just need an email address and a password, and you are set. It's really user-friendly and has many training materials, videos, support, and yes, a really great community. no programming needed for the usage of Galaxy, and it enhances reproducibility in science from this so-called fair data. I will talk about this in details later. And just a quick note that where I put this computer icon, there's a link available for you to see it or check it later. For Galaxy servers, there are basically 3 main servers that everyone uses. The American one, the European one, and the Australian one. But there can be hundreds and hundreds more local ones. For example, the Semmelweis University has also a local server where you can put tools, whatever you want to include in your server. For example, if you use only protein structure data, you will include only those tools, but you will use for this kind of analysis or imaging or something else. So it will be more effective and fast in your server or in the cloud. As this main page of Galaxy suggests, Galaxy is really useful for scientists, for teachers or trainers, for tool authors and developers. And it's, of course, maintained by administrators. Okay, but where you should start? I think you can or you should check this link, the Galaxy Community Hub, where you can get familiar with the terminology. There's a glossary, tutorials, events. You can join the community and get help or ask or read the FAQs. And meanwhile, I really suggest to check this GTN, which is the Galaxy Training Network. Basically, this is the heart or the brain of Galaxy, because there are lots of slides, hands-on videos, help, full materials uploaded in this web page from beginner level to advanced level. And there is an annual or biannual event or course, as you can say it, which is the GTN Smorgasbord. I don't know, I can speak in Swedish, but it's held in every year or every 2 years, and it's free and it's online. Usually takes 5 days, and it basically tells you everything about the specific tools in Galaxy. You can learn with the administrators, with other learners, with other people, scientists, and there's a Slack channel available 24/7 if you want to ask anything from the experts. And the Galaxy project has a YouTube channel also, but I didn't really check this before because I think there are more material and videos in the GTN platform, but you can check it if you want to. Some quick words about Galaxy training in Hungary. As far as I know, only University of Debrecen and Semmelweis University have Galaxy training for postgraduate and graduate students. And if you or your university have these courses, please intervene, and I will modify this table as soon as possible. But I think there are only 2 universities now. And if you are interested in bioinformatics and Hungarian bioinformatics, I've put some links here that you can get familiar about the groups, about the conferences, training materials, etc. So let's see the Galaxy itself. It can be divided into 3 parts. On the left side, you can see the tools. In the center, there's the main page. And on the right side, there is the history. We will check each one of them. Firstly, the tools. In the tools menu, first, you have to upload the data that you want to investigate. You can do this with by many options, either as a get data, upload data, or import from another one, history. I will talk about it later. The get data option is connected to the big bioinformatics servers such as NCBI, EBI, and the others. And you can write the IDs or the links, anything in here, and it will upload your data set into Galaxy. The other option is the upload data, which can be this regular one or option. You can upload from your computer. You can use FTP servers to upload bigger amount of files. For example, from 1000 Genomes projects or the TCGA database, or you can add the URLs directly into this part, and it will download it. If you want to analyze multiple data sets as one, because you have many data and you will do the exact same setups and analysis on them, then it's advisable to use this function, because then your multiple data sets will be acted as one, and it will be easier to handle with the downstream processes, and it will be easier to manipulate your data sets. There's another option, what is called the so-called rule-based option. And in this part, you can modify, manipulate your files just like you do it in an Excel program. So you can rename the columns, add, remove, basically do anything with this, and you will have a cleaner input data set. If you are not sure what data upload to use, this is a really good flowchart. I found it in the Community Hub platform. And now let's see the tools themselves. This part is called the tool shed, and these are the tools. There are currently more than 9,000 tools available in the tool shed, and they are public and open for everyone. And they are being developed and maintained, updated by the community contributors every day. So, but there are, I have checked it yesterday, and there are a lot of outdated and not so useful tools. So I think it's much more less number what you actually use. Yeah, and sometimes you have to install it via your terminal or a programmatic access. So it's not really that simple to just click and use, but it can be managed. So we have seen these tools, and I have put this screenshot about the very, very wide variety of areas that the tools cover. So you can use it for epigenetics, imaging, machine learning, genome assembly, microarray analysis. So yeah, a lot of options. But I won't go into details about this because I don't think that's the scope of this lecture is. The main part is the center part of the galaxy. You see news and some informations when you click on this, but more importantly, when you open a tool or a history data set, you will see the actual setups that you want to use in your analysis. For example, here I used this column arrange tool, which is a really simple tool where I can insert columns by specific columns name. So yeah, just rearrange the columns, but I used it as an example for today. So yes, from this input data, I got this output data. And the other really important part of Galaxy is the history. Your history is basically the working directory where you can find everything what you were working with, your input files, whether they are updated by you or imported from someone else's history. You can see all your computational steps with given parameters and every output files. So yes, it's your history. And at once you can see only one history, but you can have many other ones in your account and just easily switch between them. You can have empty history also, what you can hide or delete or reuse again with new data sets. You can do the same with your data sets also, either as individual ones or multiple ones. I mean, you can select them or select all to hide a specific data set, to delete or purge, which means delete it permanently. You can build data set lists or pairs, which is, for example, when you have a sequencing data from a reverse and a forward read, you have to merge them into a pair data set. And I'm sure there are other options that someone else use. You can hide the specific data sets that you don't want to see, and you can check it what you have hidden before. And the same is true for the deleted data sets also. So it won't be deleted, of course, immediately. You have to click on this permanently delete files to remove from the server. So after we clicked on the run tool or the execution button, you can see these different color codes on your history, where the green one means that the job is done and it's completed. The red one means an error, either by your mistake or because of a bug. The yellow one means your job is still running, it's not ready. The blue one means it's paused by you because you don't need it yet, or you just want to fasten the analysis. and if it's gray, it means that it's still waiting for its part in the server, because the computational capacity is not enough. So yeah, many times it's gray because you have to wait, unfortunately. And the really good part that you can share your history with specific users, with everyone. You can send it via link, via an invitation in email. So yeah, it's just really cool. I wanted to show you some other parts of Galaxy, but most of the people might not use that so much. One is the workflow, the other is the visualization, and the third one is the pages part. I don't think I need to explain this too much, the visualizations part, because you can check what you want to use, which tool you want to use. For example, you can check your genome, like in the IGV browser. You can make box plots, Venn diagrams, heat maps. Cytoscape is already in the Galaxy. So yeah, it's also really convenient, and you can also share and publish these, download the image file or PDF file, or export it. There are many other options, not just this, what I have screenshotted. The other thing is the workflow. It's also really useful when you have multiple sets of input data, because you can use them as one single file, basically, and you can give them all computational steps, parameters, and it's basically kind of like an iteration, because you just reuse the same steps in every data sets. And you can also redo someone else's analysis with just like 4 clicks if you have his or her workflow. I will show you an example. On the left side, you have again the tools, which you just drag and drop here to the graphical part. I don't know the exact name, but I call this the graphical part. And the example that I showed before was that we arranged some columns in a tabular file. So now I can just put this input data set. I click here, and I drag this box here, and I give a name, and I show that what I used in this data set. And on the right side, you can set the parameters just like in the previous slide. So you basically do the same steps, and do the same like in a normal tools program. But it's really good because the same jobs will be executed and put in your history, and you don't have to do it every time to upload your data set, to upload hundreds of data sets or thousands. You just do this workflow at once, and you will reuse it months or years later, or you will use someone else's workflow on your data set. So yeah, it's really good. And you can also share it, publish it, download it, export it for publications. and it looks really good and cool and clear. But it's not just a good visual image or a workflow. It's actually a file that you can use again in your analysis with your own data sets. And the third one is the pages part, what is connected with your histories, your workflows and data sets, and can be imagined as a lab notebook, or if you use R, it's a kind of like an R markdown. And it's the annotation of your whole experiment. Just like in your workflow, you can drag and drop these tools or these parameters. For example, which data set you used. You can upload images. You can upload your workflows. Set the current time and the Galaxy version, and add visualizations inside. So you don't have to write these. It's just how it translates these clicks of here. And the output will be something like this. It's again just a really simple example where I put the title of my data set, my name, the Galaxy version and date, the data set I used, and the linear graph of this data. And you can also share it, publish it, download it as an image or a PDF, depending on the file that you created, export it. So it's really good for yourself, for other scientists to reuse your analysis. It's really good for publications because the figures and the graphs are really good and high quality, and you can put this whole annotated description in the supplementary material. So yeah, it also increases the reproducibility of your work. And that's the last slide of my presentation. I hope that I could show you some parts of the Galaxy, that it's really, really a good platform for data analysis, but it's not only this, because it's a really good data management platform, can be used for many visualizations, and for publications to make your work reproducible, and make some nice figures. So yes, I want to thank you for Tamás Kis, the invitation to give this presentation for you, to Balázs Györfi, who is our PI in our group, and to Beata Scholz, who first introduced me Galaxy and taught me it really well. And of course, I want to thank all the participants for their kind attention, and I'm waiting for your questions. Thank you.
Thank you, Szonja, for the informative, great presentation. Now it's time for a question. If you have any, please type it to the chat window of the Zoom. While you type your first question, I already have one. Szonja, so we all know the GDPR rules are really, really strict. What happens if I have patient data? Can I upload them to Galaxy? Are these data sets safe up there? Can I analyze it for my next publication?
Yeah, it's a really good question, because patient data are very sensitive in everywhere in the world. So actually, I have never used it. I mean, the clinical or personal data in Galaxy, and I'm not sure it's that platform to handle this kind of data. But of course, the servers and the IT, the technical part is ensured to handle such data. But I'm not sure that personal data should be uploaded.
I see. But I guess there is different when you use the global server and Semmelweis uses their own server, right? Yes. Thank you. So.
Thank you.
All right. I think we have a question.
I don't see.
Okay, then I will.
Now. Okay.
Okay.
Does it require any institutional email for? No. I use my Gmail account.
And we have another one from Sarah. It's directed to me. So the question is, does the tutorials contain a workflow too, so I can immediately run it with my own data?
I'm not sure about the workflow, but there are these so-called hands-ons where you can, upload your own data, and there is actually step-by-step guides for this. So yeah, you can use your own data. You're welcome.
I also have a question. What is there some kind of data limitation, size limitation, what we can upload there?
There is a limitation in every server, which is not so high, I think. If you use, for example, sequencing data, which can be really big, or some images. I don't use this image data, but yeah, there is limitations, and sometimes it's really struggling to think and delete your files that you are sure that you won't need in your later steps, or you just go on another server and continue your analysis from that part. So yeah, it's not that good. But if you can or you are good at this, you can make your own server or domain and increase the capacity.
So maybe this is the downsides of the freely available service, right?
Yeah. And you have to wait a lot of, because there are many users from all around the world, and I don't know the exact number and how many people are using it at a specific time, but you have to wait sometimes a lot for your part, for the queue to be available for your job, the analysis job. Yeah, it's sometimes slow. I will check the questions. Get data from public databases and out on data may require quite large storage, and also we need CPU time. how is it managed? You can use the FTP servers for these larger data sets. So you don't have to download it for your own computer. And there's a connection between bigger bioinformatics databases and Galaxy. Here. Where did I put? Yes, so I don't know the exact IT behind this, but of course it doesn't download to your own computer, and it doesn't use your own CPU or capacity of your computer, but it uses the server's CPU and computation to download the data into your account. So I think that's the answer for your question. What interface can be used to register for the May webinar? Sorry, how do you mean interface? You just need a Gmail account. I don't know if you need a Galaxy account to register. I think, yeah. I didn't register it without my Galaxy account. So yeah, it just it's really easy. You don't need anything special. Do these servers use some quota system? Is there information on how to establish local Galaxy server on our own? Yeah, they use some quota system, but I don't know how, and yeah, the technical details. Yeah, I can give you a contact of the Semmelweis Galaxy domain if you want, about how to establish a local server. And yes, there is information about this how to do it and so on. But I'm in the same group with the ones who did the Semmelweis server for Galaxy. If it's okay with you, I can give you the contact. Cool. I don't see any other questions, but please keep asking if you have any.
All right. I think we can end this webinar soon if you don't have any further questions. Szonja, I think she will be available later via email, so you could see her email address on the slides. So I think
yeah, it's here.
Yes.
But
so yes. Thank you very much. So thank you, Szonja, for the great presentation, and thank you for everyone for joining us today. Yeah. Have a nice night.
Yes,
Bye.
thank you very much, everyone, and Tamás.
You're welcome.
Can we get the slides? Yeah. Yeah. I will send. Thank you. You're welcome.